Welcome everybody to the 12th edition of the Multiomics Reading Group. I'm your host for the day, Cesar Miguel Valdez Cordova, and today we have the honor to present Dr. Xiaojie Qiu, who will be presenting spatiotemporal modeling of molecular holograms, which is now published in Cell. For a brief introduction, Dr. Xiaojie Qiu is an assistant professor in the Department of Genetics within the base program at the Department of Computer Science at Stanford University. He worked with Dr. Cole Trapnell at the University of Washington during his PhD to develop monocle 2 and 3 for pseudotemporal trajectory analysis of single-cell RNA-seq data. During his postdoctoral research, he collaborated with Dr. Jonathan Weissman, initially at UCSF, and then at the Whitehead Institute and MIT, where he developed Dynamo, a tool for reconstructing RNA velocity vector fields and making reprogramming and in silico perturbation predictions using metabolic labeling-enabled single-cell RNA-seq. Recently, Dr. Qiu also led the development of Spatio, an advanced tool for spatiotemporal modeling of single cells. His lab is currently focused on predictive modeling of single cells through a unique synthesis of genomics and machine learning. Without any further ado, please, Dr. Xiaojie, take it away.
Thank you. Yeah, thanks for this very nice introduction. Yeah, it's my great pleasure to participate in this seminar. And for today, actually, I'm gonna change. I'm gonna talk a little bit different from what I proposed, and actually wanna talk about how to move towards creating predictive watch embryos.
Wonderful.
Yeah, so I want to motivate my talk by asking just a simple but fundamental question. How does a single fertilizer cell give rise to an entire organism with trillions of cells and in a precisely and spatial temporally controlled manner? I think many of you probably already know, thanks to the development of the single-cell RNA-seq, nowadays we now able to generate millions, even 100 millions of single cells. And institutes like CGA or consortia like Human Cell Atlas now propose the idea to leverage all just public single cell data to build virtual cells, to have a virtual representation of the cell states, and then use this states for a lot of downstream applications. So I think this kind of idea is definitely very exciting, has a lot of potential, but I always find that it's not ideal because, as we know, it's also shown in this figure, the cells actually lives in a 3D volume. They talk to other cells in the 3D space. So the spatial niche matters. And also cells is changing, they're moving over time, both in the physical space, but also in the cell state space. So ideally, what we really want is to move away from just isolated, static virtual cell concepts to more like intact, more like dynamic, systematic organ level or even embryo level modeling. So one of my main goal in my lab is to create virtual organs and even virtual embryos. This is obviously it's a very ambitious goal, right, to creating a virtual organ or virtual embryo. But I think we have a great strategy in my lab to achieve, hopefully, this ambitious goal. So we will start with experimentation and then computation and then doing some predictions. So in my lab, we have a wet lab component we are able to implement and also even develop novel single cell spatial assets, and then we can use this technology to generate multiple single cell spatial data sets. Once we have such a data set, then we can use it to build interpretable machine learning models to understand the underlying gene regulatory network, and then we can use this model to be able to gain mechanistic predictions, such as like the prediction of the congenital heart disease and many other diseases. So in the next few slides, I'm going to actually walk you through some of the work I have achieved in the past few years that tries to achieve this virtual emperor goal. And I'm gonna start with talk about some of my postdoc work very briefly, and then we'll focus on the the special package that originally proposed for this talk. So to model the spatiotemporal dynamics during my postdoc, and developed a framework called the Dynamo that tries to reconstruct the RNA velocity vector fields to model the temporal dynamics of single cells. And as probably many of you also know, the concept of the RNA velocity, right? So the RNA velocity basically is a computational method that leverages the spliced and unspliced RNA to estimate the temporal dynamics, basically instant change of the transcriptome states. And that was previously based on the spliced and unspliced RNA, which is quite biased because when we design the single cell RNA-seq profile approach, we didn't aim to capture the unspliced RNA. So in my postdoc, we implement the method called the 10 resolved single cell RNA-seq, basically using RNA metabolic labeling to explicitly tag the next RNA to separate the new RNA from old RNA, and then we then use this method to be able to reveal the cell temporal dynamics. And once we get the RNA velocity, the machine learning problem now is how we can take paired gene expression state X and corresponding velocity dx over dt for many cells to learn a mapping from the expression state X and to the velocity dx over dt. And you can achieve this using in the original paper using a Kuhn method, but we can also use a neural net. But the key thing is this function actually have deep biological meaning. It actually corresponds to the underlying gene regulatory network, basically how each gene interact with each other in a single cell. Once we have such a mathematical representation of a single cell, then we can use it to predict, for example, how each individual cells evolves over time in the gene expression space. So in this case, as shown on the right, where we give some initial state of the hematopoietic stem cell, and then the SHC cells will evolve following the vector field function to first become GMP, MEP-like cell, and then ramificates into all kinds of terminal mature blood cell types. And if a fundamental innovation of this approach comparing to the most of the existing single cell analysis approach is that it's one of the work that enable us to make non-trivial predictions and also gain mechanistic insights. So for the sake of time, I'm going to here just quickly tell you how we can achieve gain this mechanistic and predictive insights. So on the one hand, once we learn the vector field function fx, where we can actually calculate the Hay order derivatives, we can calculate the partial derivative of fx respectively to the state variable x, which gives you the Jacobian. Basically, if you see changing one gene x1, for example, and then the velocity of another gene x2 is also increasing, then this indicates activation and vice versa. So using Jacobian, we are able to recover the underlying gene regulatory network. So here, as you can see, where we can use this to recover a minimal network explaining the emergence of megakaryosis. And this is something like people never able to figure it out for the past decade also. The other thing is that once we have this vector field function, we can use it to calculate the least action path, which is a key concept in physics, and use this approach to predict the optimal path that convert one cell state to another. So basically, what we are doing is we calculate a path integral following this function, where the here v is the velocity, along the path, the fx, that's the vector field function, where d is some diffusion term. Normally we just set d to be a constant, and then we can integrate this along the path under some time limits, then to get the action. And the goal is to minimize this action such that the transition from one state to the target state has a minimal action. So using this method, we are able to identify the optimal path that convert from one cell state to another state, and also identify the transition factor cocktails that enable such a transition. Lastly, we can also use this approach to make in silico perturbation predictions. For example, we can use it to simulate the whole. If you lock out the P1, how the cells will respond to such a lockouts. And this mathematically this is quite simple to achieve. You can just multiply the Jacobi matrix by the delta x, which is the distance of the change of the gene expression, basically the perturbation. And in this case, you can see where the after locking out the P1, the cells from neutral field monocyte, they actually move away from its current state and move towards to become MAP-like cells. And this makes a lot of sense because the P1 is a master regulator for GMP lineage. So if you lock out they will actually move to the alternative MAP lineage. So I have quickly, briefly mentioned that using this dynamo framework, we are able to gain mechanistic insights for the temporal dynamics of single cells. But as I mentioned in the very beginning, right, the embryogenesis and essentially for most of the biological process has also the spatial dynamics as well. To be able to enable modeling along the spatial axis, just as what we did before for the temporal dynamics, where we also need to model we identify the metabolic labeling enabled 676 is ideal technology to generate the data for the temporal modeling. So for the spatial dynamics, we also need to identify the basic technology that enable the spatial modeling. And just a quick background. So the existing spatial transmitomics methods, like the 10x region and others, they have actually very low resolution because each spot corresponds to a lot of cells, and the distance between each spot is really far apart. And so this certainly cannot give us subcellular, even single cell resolution. However, about 2 years ago, I collaborated with the BGA, and we reported this method called Spatiotemporal, and which is a sequence-based technology. So they actually reproved the sequencing chip to do the spatial transmitomics profiling. So on this chip, you have a lot of billions of DNA nanoballs, and the size of the DNA nanoball is very small. So for each cell, you can actually capture up to 400 DNA nanoballs. So this essentially gives us a subcellular resolution. And another unique feature of this technology is that it has a very large field of view. It can capture up to 13 centimeter by 13 centimeter field of view. So you can easily put like an entire mouse embryo or even a adult human brain on a single chip. So this was really like a breakthrough technology back then. And in order to demonstrate the power of this technology, we then used it to perform, to create the first spatially and temporally resolved cell atlas of mouse organogenesis. So we collect the mouse embryo from like embryonic day 9.5 all the way to 16.5. And at each time point, we collect about 2 to 3 sections on average. And then we profit with the stereosheet. Then we can computationally analyze each individual cell, and then we can map just the cells back to the physical space, and we can color the cell based on the organ. So you can see where we now have about 30 or 40 different cell types and organs on this across the most organizations. And they are special organization with very dynamic and also very intricate. If we zoom in into individual organs, like the heart, which is the red, this red circle here, you can see over time it evolves and it dramatically changes. That indicates the heart is like going through the organizations become more functionalized. So as I mentioned, this was like a breakthrough back then. So it is actually this work was published as a cover story, but there is a very obvious limitation, which is we only did about 2 to 3 sections per each embryo. So the so we cannot really capture the entire picture of the mouse embryo, right? Um, Ideally, if we want to understand embryogenesis, we would love to know the entire embryo, and then this is exactly what we did in the past 3 years or so. So we in the past 3 years we moved away from the 2D cell atlas to create a 3D home mouse embryos, and we developed the associative computational framework of the spatial. So spatial is actually a very general, comprehensive tool for spatial transmetabolic data analysis, and is especially toward for doing the 3D level spatiotemporal modeling. So it starts from taking sequential 2D slashes to reconstruct the molecular hologram or 3D maps of the organ or the embryo. Once you have this 3D map, then you can use it to do a lot of innovative downstream analysis. Like you can identify the spatial axis, yeah, along the AP axis, anterior, posterior, and the dorsal ventricle axis, and identify genes that change along these axes. And we can also try to model the cell-cell communication, even like the communication at the tissue level, because we now have the 3D embryo. And I'm going to also talk a little more extensively on how we can use this method to actually connect the cells across time to predict the cell movement in the physical space. And we also developed like a tool, like a viewer to facilitate the data exploration of the massive data sets. So this work was published in Cell and also recently highlighted in Nature and Nature Masters as well. So this the experiment is actually quite tedious and very expensive. It actually cost our collaborator more than $1 million. And the workflow is actually, but our workflow is very straightforward. What we did is we take the mouse embryo from embryonic day 9.5 and also 11.5, and then we perform the continuous cross section at a depth of 10 micron for most embryo at embryonic day 9.5, because the embryo is relatively small, so you can actually perform continuous section. And then you can profit with the stereo-seq for every sections. And for 11.5, the embryo get much bigger, so we cannot really afford to do continuous profiling with the stereo-seq. So what we did is we for every 2 sections we did the stereo-seq profiling, and then we collect the correspondingly the broad field image for the same section for the stereo-seq profiling, and also the HE staining for the consecutive slats for the stereo-seq slats. So after some data cleaning up and filtering, in the end, for the 11.5, we have a total of 85 sections. And one obvious thing you can observe from the data is that now because we put the tissue section randomly on the chip, right? So the relative position between tissue section is gone. Therefore, a very natural question, first question is how we can actually reconstruct the 3D embryo model from the sequential 2D slashes. It turns out the 3D reconstruction is not a trivial problem. It's actually very challenging. So, for example, this is because there are a lot of issues involved. For example, when we do the tissue sectioning, the tissue slice may experience different force. So this will cause non-rigid deformation. Secondly, when you do the tissue sectioning, because of like the experimental error, sometimes you may introduce partial overlapping. Like only part of the tissue embryo is able to share. It's aligned across consecutive tissue sections. And the modern spatial technology is very actually has a very large data sets. For example, for the most embryonic day 16.5, we have more than 200,000 single cells. For the most cortex dataset, we have more than 500,000 single cells. There are also other issues like the error accumulation, G-shift problem, and the multi-modality problem. So for error accumulation, because we have a lot of sections, right? So if you alignment, especially if you use like pairwise, sequential pairwise alignment, like every time you only align 2 sections, this error from this pairwise alignment will propagate across sections, so leading to large shift in the final sections respected to the very early ones. Secondly, the alignment also tend to center align all the tissue sections. But the but sometimes the tissue section may have some the curvature, and so this is a G-shift problem. So also for the spatial data, often we not only have the transformational information, but we also have the imaging information. So the question is how we can actually integrate both imaging and also spatial data to improve the alignment jointly. In order to address all these challenges, we then developed a generative Bayesian framework to achieve the alignment of the tissue sections. So here we have let's say we have two samples, where sample A, sample B. We assume the sample A or the slice A is the model point. The sample B is the data point. So the model points can go through some reached or non-reached transformation to generate the sample B. And then we can write down the likelihood function of this this mixture model. So we assume there are like outlay. So this will handle the partial element, and the rest will be like the inlays. And we can formulate the generative probability as a function of the gene expression similarity and also the spatial proximity. We can also additionally incorporate some like prior information, like this outlay model, the probability of gamma is full of the beta distribution, and so on and so forth. Then we can write down the full joint positivity distribution shown on the red. And this then we can use a maximal a posterior to identify the optimal parameters in the searching space that gave us the highest likelihood to generate the sample B from sample A. Once we have this optimal the parameters, then we can use it to recover the generative probability and also the transformations. So in order to enable our alignment works at the embryonic scale with 3D multimodal data, we then develop a series of extension and the optimization. So we first implement the GPU acceleration. We also induce some variable approximation and also stochastic variational inference. as well as like sparse matrix calculation to further enhance the efficiency and scalability. Second, we also extended the Paris element to multi-stage global optimization to just address the error accumulation issue mentioned before. To address the G-shift problem, if you have the mesh, like for the most for the most brain rare, you have the N Institute common coordinate system. You can use that to do most to do the mesh correction. This will help us to address the G-shift problem and incorporate some shape information. Lastly, we also implement the method that extract the image feature and enable imaging cadence when we do the alignment. So here I'm gonna show you an animation on how the spatial partial element or non-restricted element works. So on the top and the bottom, just like two tissue sections here, and the point, of course, are cells, and the names are the corresponding the maximal element. So you can see initially its algorithm will try to like for the non-restricted element, it will rotate the translate, but then we have some like local shifts. For the partial element, you may see you like just in the inner probability, the red color, it means is inner, it's blue, it means like the outer. So there is a region in here, is like missing. So that indicates the outer layer. We also compared with existing model like PASTE2, Moscow. In general, I think our method is much better. So after I introduce the model, now let's actually take a look on the actual results when we apply this model on the whole most umbrella datasets. So what I'm showing here is a 3D map we reconstruct for the most umbrella at 11.5. So here each point is a cell, the color is the organ. And the first thing you can see from this animation is that it really looks like a most umbrella, right? It has like the brain, the nymph, the tail, spleen, and everything, right? But not only the global structure makes sense. If you zoom in into each individual organ, like jaw, liver, brain, tail, heart, nymph, right? You can also appreciate the intricate 3D structure for each organ. Also, it's also very accurate. And our method is very general. It can be applied to other technologies as well. So we previously used the stereo-seq data, but this simple can be simply applied to morphic data as well. So what I'm showing here is the 3D map of the human heart. We slashed about 53 slices and about 3 million single cells. Yeah. Because we only select the 53 slashes, so on the left, you can see there is a lot of gaps in between. So we then developed the algorithm that interpret the slashes. Now you can now see like the vessels on the surface, like just small strings, and actually. Nobody is able to previously reconstruct such strings before, and this will be really important for a lot of heart disease. So we can also zoom in into each substructure. Like we can also look for trabecular ventricle cardiomyocytes from the left atrium and the red atrium, and the compacticle VCN from left ventricle, red ventricle, and so on and so forth. To further accelerate the data exploration, we then developed a Google Earth like browser for interactive 3D data exploration and visualization. This is already released here, and you can actually explore our 3D data, 3D umbrella data if you like. I think the purpose of the special paper is not just about the 3D reconstruction. What we really want to do is to actually uh, perform some innovative downstream analysis. And here I'm going to highlight the spatiotemporal modeling. So we have the 3D map of the embryo across multiple time points. Then we can try to create a cell-to-cell correspondence. Once we create the cell-to-cell correspondence, we can then learn a vector field, the cell migration vector field. Actually, we can use the same method that we used for the 3D element. And taking the heart as the example, because the heart is already mature, it's the first organ appear during the embryogenesis, so it's already mature at this, more or less mature at this stage. So we can first create a cell-to-cell correspondence, and then from the correspondence, we can create a morphogenic vector field, and then we can predict the cell trajectories. Now the question is how we can actually quantify this morphogenic vector field to gain some biological insights. Previously, as I mentioned in the dynamo work, we showed that we can leverage some vector calculus like the Jacobian, as I mentioned, but also acceleration curvature to calculate the RNA, the regulatory network for the single cell data. Now, with the morphometric vector field, you can actually also do similar vector calculus on the physical space. So now the space is not in the gene expression space anymore, it's actually in the physical space, so it has real physical meaning. So the acceleration really tells you the rate of cell movement. And the core tells you whether the cell has a local rotation in physical space when the moving. Curvature tells you whether the cell make a turn during the cell migration. Divergence tells you whether this is the source or sink of the cell migration. Torsion tells you whether the cells, the 3D object has twist during the movement. Jacobian tells you actually if you change once you move in one dimension, whether it will actually influence the other dimension. And using the 3D enablement method, we can actually analytically derive all these quantities. So the calculation of the differential geometry is really efficient. So we then use this method to calculate the morphogenic acceleration core and divergence for the heart at the embryonic day 9.5. We can calculate just each property for each cell. Showing here, where as you can see for the acceleration, you can see the red artery has the highest acceleration. Where other regions has a very low acceleration, and this makes a lot of sense because the red artery it comes from the second heart field. So to comparing to the first heart field, like with ventricle red artery, the red artery from the second heart field is actually still developing. So that's way it shows stronger accelerations. Similarly, we can also find the red ventricle or left artery has a more stronger core, indicates more stronger rotation at those regions. And for the divergence, it shows the red ventricle has the strongest divergence, indicates this is the source of the movement. So the next question is how we can actually link just microscopic morphological changes to the underlying gene regulation, gene expressions. So we find that since we have both the gene expression value and also the spatial location, so we can, I mean the morphogenic property, we can then perform a associative analysis by regressing the gene expression respected to the morphogenic properties, and then we can identify which gene significantly change as a function of the morphogenic properties. So we then calculate this for both for the acceleration core and also divergence, and then we group these genes into different spatial domain, like the left red atrium, ventricle, or the flow tract, and then we sort the genes based on the Q value, right? And then we can we see a lot of genes indeed really like morphogenesis relevant. For example, the NGPT1, right? This is a gene that show association with the acceleration and is enriched in red atrium. In fact, the NGPT1 is a gene responsible for the antral chamber formation. So this makes sense. For the PITX2, which is a gene has a high expression in the left atrium and associates with the core, so it's related to the left red asymmetry. And for HER2 gene has a highest value in the red ventricle, which is associated with the divergence, and it has a highest. It's actually also shown previously to relate to the ventricle formation. So
Sorry.
what I mean, yeah,
Yeah, I have a question. Just to clarify one point, I don't sure I understood properly.
sure.
Your data is spatially reconstructed data at
Yeah,
one time, right?
and then 2 10 points.
So you have 2 time points.
Then you can, it's shown here, right?
So the cell correspondence is between 2 time points, not between 2 layers of your spatial
no,
data.
yeah, that's not because we want to study the
Yeah.
movement of cells.
Yeah, I agree. Okay,
Yeah.
but and so to acquire the data, how do you proceed? Because I guess like if
You cannot track the sensor, of course, over time, right?
yeah
So what we do is I mean, I think I briefly mentioned this, right? You collect the most umbrella 2 10 points.
yeah
These are
okay
different emperors, 9.5, 11.5. But we assume uh, uh, more or less all the emperors go through the same process,
okay
or at least similar process. Yeah.
okay
I mean, this is a fundamental assumption for all the single cell and the spatial data analysis. You cannot trigger the same cell over time. Every time
Yeah,
you do a measurement to kill the cell, but we can assume it's like
yeah okay
consistent
That's
across
clear.
samples.
Thank you.
Okay, cool. So I think the what I described it's actually is kind of like associative analysis. We associate the gene expression to the cell movement. So it's not a causal. It's like basically doing some post processing. Ideally, what we really want is we wanted to have a joined model. I think this will be the way to move towards building the virtual heart, virtual organ, or virtual embryo. So this joint model should jointly model the cell migration, cell growth, cell division, and cell state change. So this is actually some like early result we got for modeling the oxytal brain regeneration. So here each point is a cell. You can see cells are moving, right? And you can see the colors are changing. That indicates the cell states because the color is the cell type. And you can, if you look carefully, sometimes there are new cells come out in some regions, and the cells may be disappearing. So this indicates the cell growth and cell division. This, in many ways, is actually similar to this kind of like the live in total live imaging tracking. You can use live imaging to track the movement of each individual cell. But the advantage of what mounted on the red is that although we are not continuously tracking the same cell, but we are we predicting the cell movement. At the same time, we can have the transcriptome information, where the live imaging tracking data shown on the left only able to give you like maybe one marker, like in this case, MACE P1 gene. And we actually applied this method to simulate, to predict the hard organogenesis. Showing on the left, you can see we are able to simulate how the heart grows over time, right? But importantly, now we can do some causal analysis instead of like try to calculate the acceleration and identify which gene is associated with the acceleration. Now we can actually do some causal analysis. We can directly calculate the Jacobian from the gene expression, the cell migration velocity. So, for example, we see for the HER2 on the left ventricle region, like just blue area, at the index, the HER2 gene in this region repress the cell movement. Well, for LTGB3, it's actually the opposite. It activates the movement. So what it means is that in the future, we can actually in silico lock out some gene and simulate how the heart grows. This may help us to understand the heart defects, which will be like fundamental for the congenital heart disease prediction. I think how much time do I still have?
I'm sorry, we have 12 minutes left. And
Okay,
we would
cool.
also...
So maybe I'm gonna quickly walk you through some of the foundation model. Probably also more interesting to some of you as well. So I think what I mentioned here is like for a unique data set for the mouse hardware. Ideally, we actually want to be able to build like a universal virtual embryo model across different species, and this is obviously the ambitious goal, but now we made some early efforts along this lane for research. We created the first foundation model in my lab called Tabular. The Tabular is significantly different from existing foundation model like the SGBT gene format SG foundation. It's a privacy preserving predictive foundation model with federated learning and also tabular learning. There are three major motivations behind this model. Firstly, when we want to create foundation models, you will collect hundreds, millions of single cells, right? So it will involve a lot of individuals. And previously, we were finding that if you aggregate the single cell data from each individual, you can actually link the gene expression data to the EQTL datasets or other genomic database, and using this to de-identify the patient. Secondly, so this could cause into some like privacy issues. So we need to fix this. Secondly, the existing single cell foundation model basically tries to borrow methods from natural language analysis to process into like those foundation model for single cell data. So they treat the single cell data as sequences of genes for each cell, and then try to use auto-regression or mass regression modeling to model the to do the pre-training. But the single cell data is actually fundamentally different, right? Each cell, I mean, single cell data basically is a matrix, right? It has rows per cell, columns per gene. So it's a matrix, it's the cell per gene matrix. And the order of each gene in each cell doesn't really have a meaning. You can commute the gene order and it's still the same cell. And also each gene has not only a name, but also the gene expression value as well. And the value is often very noisy. So all those features made it unsuitable for pre-training the foundation model, just by copying like methods from an NLP. The third thing is it's actually related to the first motivation. So a lot of labs and institutes wanted to build the foundation models, but we don't really have, most of us don't have the huge amount of computing. So ideally, we want to be able to build this model in a distributed way. So other audience motivations table, then we then develop the tabular. So in contrast to existing foundation model that aggregate the data to a central server and train the model on the central server, the tabular model tries to train each provided data sets locally in each institute or each hospital. But then they're gonna upload the data in the central server and then aggregate it there, then update the model and back and redistribute it back to each institute. By doing so, you are doing essentially doing distributed learning. So the training is much easier, less computing intensive. Secondly, it is still, although it doesn't share the data, it still benefits the data from other people because we do share the weights. So this addresses the privacy concern. Secondly, we design a very unique pre-training strategy for the single cell data. We realize the single cell data are more like a table, right? So we perform tabular learning. And for tabular learning, we start with creating a corrupted view of the gene expression matrix. By basically randomly select some cell for each gene and then sample the gene expression value from the marginal distribution, the negative binomial distribution of this gene across the cells, and filling in the sampled value for the selected cells for this gene. And this will create a corrupted view of the original data. Then we can try to do gene-based learning and cell-based learning. For gene-based learning, we try to reconstruct the original gene expression from the corrupted view. For the cell-based learning, we actually use a contrastive learning strategy, where we in the latent dimension for each cell, it's coming from the original view or the corrupted view. We can form some positive pairs and negative pairs. For positive pairs, use the low dimension embedding from the same cell. One is from the original value, the other is from the corrupted value. For the negative pair, we just are coming from two different cells. So so the pre-training takes consider both the rows and the columns, and we found that using this unique strategy, we are actually able to dramatically improve the learning performance. So we show with only half of the data, we are able to beat all the existing foundation models.
So excuse me, Doctor. Yeah, there's another question. If that's okay.
Yeah, thanks, Dylan. How can you give more detail about how do you corrupt the data? I didn't understand, because it from the example here, it looks like the corrupted data is really similar to the original view. It's not like in computer vision where you crop data or rotate images and so on. So at first sight, it seems easy to make a contrastive learning that make positive sample close together and negative sample far away if the corrupted view is really close to the original view. So how do you create this corruption?
Sure, yeah. So I think briefly mentioned, like what we do is that we select in more details, like 60% of the genes for each cell, right? Or you can do this on the cell level as well. But we select the 60% of our gene in each cell, and then for each gene, for the genes you select, right, you sample a value from the marginal distribution of this gene across cells, and filling in this sum of the value.
Okay, so it's not just like the value of the gene plus minus 2.
No, you need to do you need to follow the distribution.
Okay.
I think that's a more reasonable way to do it. Yeah. Cool. I think for the sake of time,
There is another question, I think.
Sure.
Yeah, sorry for just one quick question. So I didn't quite understand you guys can hear me, right?
Yeah, I can hear you.
Okay, great.
Yeah,
so I didn't quite understand the need of doing federated learning. So what's the privacy here, right? So like you have cells, mostly you cannot identify individuals just by the cell expression.
Yeah, I think I mentioned that, right? So if you do pseudo bulk of all the cells from the same individual to create the cell type specific pseudo bulk data, right? They through public available cell type EQTL dataset, you can actually able to do linkage attack to identify which patient is associated with the EQTL datasets, and the EQTL datasets basically tells you the genotype. So you can use this to identify whether this person has HIV or mental disease, cancer, all kinds of diseases. I think that's the reason having the privacy concern. The other is a lot of hospitals, even for labs, right, they don't want to share their data because their data is they cost hundreds of thousands of dollars. They want to publish first. They don't want to share the data right away before they publish, but they still want to train the foundation model and utilize other people's data. So in this setting, it will also have the privacy concern as well.
Yeah, so you understand that. So now in the EQTL side, sort of you can guess someone's individual genotype, right, just based on their bulk expression, and then the EQTL information. But then the approach that you're proposing, and also we have research in federated learning, which requires a lot of scheduling, right? So you aggregate the models to the server, and then the server will send the models back to the clients, right? In this diagram that you show in the tablet, so the clients are different tissues, right? Different brain, lung, heart. But that takes a lot of time too, right? Because like you have to schedule the model updates per client, and then you send the models to the server, and then the server aggregate, and then back and forth, right? So that takes if you have different separate silos, right? So each silo might have a different GPU resource. The speed need to synchronize and update. So it is quite challenging in practice. So if you want to really deploy this in the real world application.
Yeah, that's actually a great question. Yes, I think it definitely be challenging for doing like if you do like a research project, probably it's going to be difficult. But I think we are actually thinking about like have a startup to actually work on this. I think the data can be. We even think about the ways to allow the user to upload the data, but upload it to like privacy protected servers, and you can on this platform we're gonna develop. You can allow other people to use your data or not, or you can just only like just share the weights. But then by doing so, you essentially still doing kind of what we showing here, like doing the centered learning, but the data is not able to accessible for other people. I think this is just a demonstration, right? But in practice, there are a lot of new things in the conceit, and it's something we're gonna work on in the future, even probably in a company.
Yeah, but I guess in this case, your model, you show that outperform SVGPT, right? It has a good benchmarking, but at the same time have half of the parameters.
Yeah. Half of the data. The parameters, I think, is similar. The parameters is similar.
Okay, so in this sense, like this model can be trained non-federated in a way, but it still outperform those foundation models. So it's not a contribution, right? You don't need to federated learning. Just how this works.
I think in the paper we showed that the federative learning is independent from the model itself. You can train SGPT, SG Foundation, Gene Former under this framework as well. It doesn't depend on the model architecture.
I see. So I guess the gain here is this corrupted contrastive learning, which is different from the pre-training of SCGPT, which is predicting next token gene expression. Right, so that's what maybe lead to the improvement in terms of the size of the data you need to train the model. So you have a better pre-training. That's what you're showing in the.
That's the idea. Yes, that's correct.
Sure. Thank you.
Yeah. So for the sake of 10, I'm gonna just give out just benchmarks. It's kind of boring, right? We just show we are outputting all the methods across multiple benchmarks. But the interesting thing is we actually now I think in my view, this is the first demonstration that the foundation model is able to gain some predictive insights. Previously, the foundation model just do simple things like as shown here, like the gene perturbation prediction, cell type annotation, gene imputation, multi-omics integration. Just simple tags, you don't really need the foundation model to do it. But here, I'm gonna do something actually significantly different from others. We wanna use this foundation model to perform GL shorting prediction to characterize the pair weights and other combinatorial regulations. Basically, the higher order regulations. So the strategy is quite simple. What we do is for the input where we upper regulate. Basically, you can increase the order of this gene by 1 every 10. And we can input this change the gene expression order to the table framework going through the transformer, and then you generate a new embedding, and then you continue increase the order of this gene again, and going through the tabular transformer again. Then you can record the target gene expression as the iterations moves on. So if you see this gene in the end, where it's much lower compared to the beginning, then you can derive this is a repression from the gene 1 to gene 2. You can do the same thing for combinatorial regulations. Let's say you have 2 genes, GALA1, FLI1, regulate the EKL F1, right? These 2 genes, using the Boolean logic, you have 4 types of regulation rules. Like either both is off, then this is off, or 1 is off, and the first one is off, and the second the target is also sorry, the first one is on, the target is also on, and so on and so forth. So you can for this setting, then what you do is for no expression, you just decrease the order by 1 every 10 for each gene, and calculate the predicted value for the EKLF, and doing this for all possible combinations. So we show using this method, we are actually able to, in a completely zero shorting setting, we are able to highly accurately predict the gene regulation for 4 systems, including hematopoiesis, cardiogenesis, neurogenesis, pancreatic endogenesis. So here you can see the dashed line is a wrong prediction. You can see majority of them like a solid line just indicates the correct prediction, and the sign is also correct. It's like positive or negative. For the combinatorial regulation, you can, for example, for this first case, CBPA is regulated by 4 different genes. So there is a total of 16 possible combinatorial rule, and we show it's like across 16 combinatorial rules. It's all correct. And overall, when you look for other cases, it's around at least 80%, around 80% accurate. So this really indicates our method is able to not only predict the pair with regulation, but also do higher order regulation. And this kind of prediction is actually different from the previous prediction, because we are actually using the biochemical data that was accumulated over the past half a century or so. And I think we really have, so this really shows the power of this method to be able to gain some complicated, some predicted insights for the complex gene regulation networks. So with that, I'm gonna give a big thanks to my lab, including Yifan, who developed the 3D reconstruction method, and also Jana, who lead the tabular framework, and also many other people in the lab. So my lab is quite new. So actually, we just started my lab about 1 year ago. And I just want to also give a short advertisement. So if you are interested in like creating virtual embryos, feel free to reach out to me. You can either consider doing a visiting or maybe potentially, the postdoc or others. Thanks.
Wonderful. Let's take a moment to thank our speaker. Thank you very much, Dr. Xiaojie, for the wonderful presentation. And we went a little bit over time, so we'll open up the floor for one question. We have time for one question. Anybody in the audience? Dylan, maybe?
Yeah, well, I have a few. So if anyone has a pressing question, go ahead. I'll stick with one that I wrote down about when you mentioned that doing 3D modeling was difficult and you needed to leverage imaging data. The first part of my question is what kind of imaging data, like specifically, is it histology, for instance? And the other part of my question is whether you've thought about using, let's say, 3D MRI images, right? So for like, let's say for the brain or even for embryo, there are sort of kind of template images for those. So would those be useful for helping stitching the 3D reconstructions?
That's a great question. I think that's also something we thought about. I think when you do the alignment, you always assume the alignment, the slice is center aligned. Well, like here, when we reconstruct the 3D, so basically, if you know this field in computer vision, there is this banana problem. So like when you align, you cut the banana and you align it, then the banana becomes straight instead of like curved, right? So that's the problem. So if you don't have a 3D mesh, you're gonna lose this curvature. I think using live imaging as a like just lead sheet imaging that gives you the 3D volumetric structure, or using MRI, all things should all works. I think one of the collaborators actually trying to extend this method to integrate with MRI data.
Yeah, good. Okay, I didn't know about the banana problem, so that's good to know. I like the name.
Yeah.
Thank you very much.
Thank you,
Professor Lee.
Yeah. So if I can ask one more question on this slide, and actually the slide before, it's a little bit more of a technical question on the probabilistic model that you have for the Bayesian model. So you have this likelihood, right? So now the G are the latent variables, right? And then X are the observed gene expression. Is that correct?
X is gene expression. G is the spatial location.
Okay, so spatial location is also observed. So both X and Z are observed.
That's right.
Okay, and now you have this P of X. So this P of X is a marginal likelihood. And how would you express that? Usually it requires integral. It's not computationally tractable.
You mean P here? P?
Yeah. So below there you have 1 minus gamma times P sub O X B.
Yeah.
So what's the expression for that probability?
I think this is represented by a Gaussian process model. So
I see.
Yeah, you have some like the mean variance.
I see. So in here the latent variables are the C's, right? The Cn and the So those are you express it as.
That's right. What we wanna learn is the so the T, right? T is the transformation. C, I think I kind of forgot. C is the C. I think it's the parameters for related to the T.
So T is the alignment that you want to
T is
infer.
the rigid or non-rigid transformation. So it's like the matrix, right? For when you do the transformation, you have the transition, the rotation or translocation or scaling factor. That's just parameters.
I see. Okay, maybe I'll read your paper more carefully to understand more the details.
Yeah. This part is really intense and mostly coming from so basically we worked on this for like more than 1 year to get this right.
I see. So then later you mentioned you use variational inference and then variational autoencoder, or maybe not.
No, we don't use variational autoencoder. We use a variational inference. The variational inference is to speed up the calculation. Also, we don't have we don't really have an analytical solution for the optimization, so we approximate it.
I see. Well, the map is already kind of a point estimate. It should be pretty fast already, right? Because you mentioned you have this maximum a posteriori, where you just directly maximize your full joint posteriori.
Yeah, I think this is where the variation inference comes in. When we do the optimization, we use variation inference.
I see. So you assume there is a variational distribution for the C's, for the lane variables, or either C gamma or T, right? Those are the lane variables that you have.
Yeah, that's right.
I see. So this work you compare with the paste and then Pascal, which is the optimal transport based approach. So that approach sort of performed worse than your method, but the goal is very similar, right? So in here you also want to infer that transfer plan. Is that right? Or maybe it's the objectives are a little bit different.
It's a generative probability. Yeah, it's basically you can simulate to the transfer plan, but of course we don't use optimal transport. The calculation for optimal transport is often very time consuming.
Right.
Yeah. And you can see, this region tells you the outer layer, right? You can see it matches very well in our data, but you can see juice, you can see this region is not an end, right? This region is also not an end. I think so. Our method, in many cases, we did extensive benchmark. I think we always, at least in our hands, they were always hitting others. Another thing is we are much computationally efficient. So our method cannot even work on more than 100,000 cells, but we are dealing with 8 million single cells. Yeah.
Let's see. So these methods also they do pairwise alignment, right? So you take
exactly to do pairwise alignment. So they don't do. That's actually a good question, right? So we do a global alignment. Although we initialize with the global alignment with the pairwise alignment first, then we do the global alignment.
Got it. Right. Thank you, Dr. Qiu.
Of course. Okay, thanks for all the wonderful discussions and questions. Yeah, if you have any questions or any ideas, or if you have any friends maybe interested in my lab, please definitely reach out. And it's a new lab. We're always looking for new business students and also collaboration opportunities.